To all of you, myself Anshupriya, and in this video we are going to see about ribosome. So let's start. First we will discuss about historical background, and then we are going to discuss where it is present inside the cell, and then about the structure of ribosome and its types. So let's start. In this historical background, we will start with Claude. Claude was the first one who discovered this ribosome in the cell cytoplasm, okay, and he named it spherosomes, right? Next one is your Robinson and Brown. Robinson and Brown basically isolate, or you could say found it in the roots of Vaccia fava, and Palade termed this structure as ribosome. That's why this structure means ribosome is also known by another name, that is your Palade particles. Clear? Another we are going to see its site. Ribosome is found in both type of cell, prokaryotic cell as well eukaryotic. Another organelle where ribosome present in the cell, that would be your mitochondria and your chloroplast. In case of cytoplasm, it is freely present. There is no foundation, means there is no attachment of particles. It's not attached with any of the membrane. It is freely present in cytoplasm. Another one is your bound state, the two organelle that is in the cell, and this is basically of your eukaryotic cell. This is organelle of eukaryotic cell, since prokaryotic doesn't contain this type of organelle, right? That's why this bound state is present in the eukaryotic cell organelle, that would be your endoplasmic reticulum, and next one is your nucleus. Clear? Now we are seeing about the type of subunit, and here we are going to see two types of subunit, right? First one is your larger one, means larger one, this one, and next one is your smaller subunit, okay? This larger subunit attached to the membrane means this is bounded ribosome, and this would present in the endoplasmic reticulum surface and also nuclear membrane surface, right? It is basically present in your eukaryotic cell. Another one is a smaller subunit that is freely found in the, where it is freely found, this is freely found in the cell cytoplasm. Clear? So two sites there basically there are. Larger subunit are found on the membrane of the organelle, that would be your endoplasmic reticulum, and next one is your nuclear membrane, right? And another one is smaller subunit which is in free state. It is found in the cytoplasm of both prokaryotic cell as well as eukaryotic. Now next, types of ribosome we are going to discuss, and this is we are discussing on the basis of sedimentation coefficient. Here we have taken 70s and one minute, another one is your 80s, 70s and 80s. Here S refers to what? Here S refers to sedimentation coefficient, okay? This one is sedimentation coefficient, means the process by which, or you could say the rate at which this both larger and smaller subunit gets combined to each other, that would be your 70s. Clear? And the rate at which larger subunit and this smaller subunit get combined, that would be your 80s, okay? And this 70s has further subdivided into two types, that would be your 50s and 30s. Means this both 50s, that is the larger subunit, 30s, that would be the smaller subunit. This both subunit, when gets combined, when gets joined, the rate at which they join to each other, that would be 70s. And while when they get separate with each other, then the rate would be larger subunit, the rate would be 50s, and smaller subunit, the rate would be 30s in case of 70s ribosome. Clear? And this 70s ribosome basically found in your prokaryotic cell. Apart from this, in the eukaryotic cell organelle, that is your mitochondria, and next one is your chloroplast. Another one, in eukaryotic cell, it is found in case of eukaryotic cell, it is in case of eukaryotic cell, it is found in 60s and 40s, that is larger and smaller subunit, basically 80s type in the cytoplasm of eukaryotic cell it's found, right? And this 60s means larger subunit and 40s, they combine, they both settle down at the rate of 80s, right? This 60 and 40 is not the sum. This is the rate at which after separation this of this 80s type, the rate at which the larger subunit gets separate, that would be 60s, and the smaller at which 40s. Clear? So let's end the video. Thank you.